So adaptive immune response part 1. So we've been talking about innate, right? What you're born with, what you're naturally going to do. Your cells are going to naturally be able to detect general things about general invaders, and they're going to respond accordingly. They're going to eat them up, or they're going to ask for help. Adaptive, as the name implies, adapts over our lifetime. So you're born with it still, right? But it's going to continue to adapt and develop throughout your life. And this is the most effective means for eliminating an invader. The problem with this is it takes time to develop. And sometimes you don't have that time, right? So it takes a week or more from the first time you're exposed to that specific pathogen for it to be able to respond. In some illnesses, right, like Ebola, before your immune system figures it out, you're dead. If your immune system figures it out, though, boy, you become very popular and everybody wants your blood. Why? Because they want the antibodies your adaptive immune system has produced against that virus. So in the meantime, your innate's got to protect you, keep you alive, right, until adaptive can kick in if innate can't take care of it by itself. Right? Sometimes your innate can take care of it, no problem. We don't need to go to step 2. The awesome thing about our adaptive immunity is its ability to remember, to have memory of what it's exposed to. So we exploit this with vaccination. Vaccination, we are purposely exposing you to things so your immune system will detect it and develop memory of it. So the next time when you respond, you don't have that lag time, you don't have that wait time. Right away, it recognizes it and goes to work. And as I said, it's the most effective in eliminating. So you'd much rather have an adaptive response than just rely on your innate. This is molecularly specific, so we're talking down to individual amino acid sequences, sugar sequences on these particular pathogens. The other important component of this adaptive is that we have to be able to recognize healthy self from dangerous self, because we don't want to destroy our own healthy cells. But if they have become infected, if they're dangerous, if they've become cancerous, we want to be able to eliminate them. So there are 2 major branches of adaptive, because there are 2 major fronts of encounter that can happen. We can have pathogens that get into our cells, and so we definitely have to have that way of recognizing and detecting when cells have been infected. And so that's our cellular immunity, right, where we can defend against the inside of cells. Femoral refers to the humerus, right, the outside of our cells. And so even though some pathogens do gain entry into our cells, if we can stop them before they even get in with this femoral immunity, that helps protect us. So these 2 things aren't necessarily separate, and sometimes you'll have both responses. And so typically when I think femoral, most people nowadays refer to that as antibody immunity, because that's the major product of your femoral response that's going to help defend you against stuff from getting inside your cells, or getting the stuff that's outside of your cells. So we're talking about sometimes different types of pathogens when we're talking about femoral versus cellular immunity. And of course a different set of cells that are doing the work. So you see I even stole some of our cute little cartoons from the first webpage that I sent you guys to go read and comment on. Right? And as I said, right? This ability to remember, right? He's like, I recognize that one, right? He's telling his friends, we need to attack. That's a bad guy. And then, as I said, it's a very specific interaction. So notice we got 3 different bad guys, but only this one, particular one, is this white blood cell recognizing, right? Just that one bad guy, right? So this is the one that fits. I recognize this one. So I'm not going to play this animation. I'm going to skip over it for now. Come back to it next time. So primary response is the first time you see a specific antigen. In this process, we're going to produce those memory cells. We're going to remember this interaction. So when you encounter it the second time, your response is much faster, much stronger, much swifter. Femoral immunity works on eliminating extracellular antigens, right? So when stuff is outside of our cells. Bacteria, toxins, viruses, even at some point, right, maybe circulating through our bloodstream, our tissue fluids, we want to be able to stop them from getting inside of our cells. Cellular immunity is particularly targeted at stuff that gets inside of our cells. So viruses definitely have to get inside of our cells. So in addition to interferon, right, we have this very specific interaction for specific viruses for us to detect when cells are invaded by them, as well as intercellular bacteria, right, other pathogens that get inside of our cells. Both are very powerful. And in that aspect, they can even cause damage to our own cells if misdirected. So this system is very tightly regulated. And one of the animations that one of your classmates posted, they really go into, we're going to stick at the first receptor when we talk about in class, but it went into all the co-receptors and interactions between the 2 cells, right? Because they really want to make sure, right, that they agree that they need to move forward before they do a particular action. We don't want it to be misdirected. We want verification. We want to make sure, right, this is really what you want us to do, right? You sure? It's kind of like a computer. Most programs, right, when you hit delete, it asks you, do you really want to delete? Right? And if you actually read those pop-ups, sometimes it'll save you from doing something stupid. Your body's like that, right? It wants to make sure it's sure before it moves forward. So this is an excellent diagram showing different cells involved and generally the processes that happen. So one of our major protectors in our innate immunity is those dendritic cells, because just like our macrophages, they can engulf. But they're the major bridge between our innate and adaptive immune response, because they not only are going to gobble up the invaders, but they're going to be able to take this information to our lymphoid cells, take it to our T lymphocytes and our B lymphocytes. And so this communication that needs to happen between these cells in order to activate them, as I said, it's highly regulated. We don't want them just going ahead and being activated. You have millions upon billions of what we call naive T and B cells. Naive meaning they've never seen the bad guys yet. Right? Just like a little kid, they're naive to how bad the world can really be sometimes. They should not recognize self, normal, healthy self. Cells that recognize self, healthy self, are usually eliminated or turned off in the process of making these cells. So these cells have receptors on them for specific antigens they've never even seen before. And we've got to find a match here. We've got to find a T cell that recognizes what this dendritic cell is presenting. Once they find each other, that perfect match or close enough match, those cells become activated. And for this system, what's synonymous with activation is the next step, which is proliferation. You don't want just one cell that recognizes the bad guy. You want lots of cells that recognize the bad guy, right? So I'm hearing that guy's song in my head now. Proliferation, differentiation, right? Activation, then comes proliferation and differentiation. So they proliferate. So we get a whole bunch of them. But in addition, we need some of them to do different jobs, right? That's the differentiation. So some of them will go and do a particular job. Others will stick around and become those memory cells. That's so important in this adaptive immune response. So, and as you can see here too, right, we have two different types of T cells that we're going to talk about. We have helper T cells, and as the name implies, they kind of help out other cells. They themselves don't directly cause damage to pathogens. They just help out the ones that do. So they'll help out the macrophages and become more powerful killers. They'll help B cells become activated. Cytotoxic T cells, on the other hand, as the name implies, cytotoxic, these guys can actually kill cells. And notice we're talking about cells, cells, cells. That's what I keep talking about, right? This box in blue is our cellular immunity, mostly mediated by T cells, right? The T cells are the big boss of what happens at the cellular level. B cells, on the other hand, as you see here, are our humoral, or sometimes referred to as antibody immunity, because what's going to be produced? Antibodies. And these only work outside of cells. Antibodies cannot get inside of cells. So B cells will proliferate and they will differentiate. The ones that actually go out and do stuff are the plasma cells. Plasma cells become really big. Their rough endoplasmic reticulum gets huge because they're going to produce an antibody that they're going to secrete, which is a protein. So plasma, think of their cytoplasm, right, getting huge. And they're going to secrete these antibodies that they themselves can directly interact with pathogens, bind to the surface of bacteria, bind to viruses, bind to toxins. And then our immune system is going to respond to those antibodies binding to stuff. And we're going to talk about all the different ways in which it's going to respond and how that's helpful. So looking a little bit deeper, right, I said they're specifically interacting with each other. And as we've already seen, how do they interact? They have receptors on their surface that's going to allow them to communicate. T cells and B cells are a little bit different from each other and the type of things that they can interact with as far as antigens go. B cells have what we call the B cell receptor, or basically an antibody on their surface. And this is specific to each individual B cell. They produce their own unique B cell receptor, their own unique antibody. It has the ability to bind directly with an antigen, right? So directly with a bacteria, with a virus, with a toxin, anything foreign. And these guys actually have two sites on that molecule where they can bind. Two antigen binding sites. Unlike T cells have T cell receptors but only one antigen binding site for that receptor. In addition, this could recognize antigen all by itself, right? Where is the antigen here? It's attached to another cell. It is being presented by a cell. T cells can only communicate. Remember I said there are cellular immunity? They only communicate with cells. It has to be a cell-to-cell interaction. So even though they're recognizing a specific antigen, it's one being presented by a cell. Remember how I said there was two different major types of T cells, helper T cells and cytotoxic T cells? So each one of them has a different cluster of differentiation marker. So you've possibly heard of CD4 and CD8. Those are the markers found on helper T cells and cytotoxic T cells. CD4 is what's found on your helper T cells, and unfortunately, that is the marker that we use to communicate cell to cell, but it's also the marker that the HIV virus attaches to. That's how it gains entry. It's how it finds our helper T cells. It binds to that receptor found on those cells, binds to it, and then gains entry into those cells. That's not the purpose of CD4, though, is for HIV virus to attach to it. It is so that our cells can communicate with each other. It's like their secret little handshake. So I've used this term antigen. We defined it a while ago. So it'll review a little bit. So antigen was named as antibody generators. But in truth, they're just foreign, anything foreign to the body. Anything that the body recognizes as foreign and can mount an immunological response to. But antigens themselves can be quite complex. And especially when we're talking about these receptors on T cells and B cells, they're not interacting with the whole virus or the whole bacteria. They're interacting with small portions of that virus or that bacteria or even proteins, right? We're talking about different sequences of amino acids that make up that section of the protein. This is a very molecular interaction of these receptors with these antigens. So when we look at antigens, we actually even have to look at them much smaller, right? And we have to look at what is it that these receptors are actually interacting and binding to. So these smaller segments of an antigen we refer to as antigenic determinants or epitopes. So as I said, they got their name for being able to generate antibodies. But in fact, right, anything that's foreign that causes an immune response is an antigen. Proteins and polysaccharides can induce what's called string response, where they're repetitive molecules, and so therefore they can activate B cells pretty much on their own. We don't really need much T cell help. Lipids and nucleic acids often do not. Lipids don't have a lot of variability to them. Neither do really nucleic acids. Remember, it's just A's, G's, T's, and C's in different combinations. But strangely, not that much variation. You get much more variation when you have proteins, lots of 20 different amino acids, right? Several different combinations there. Polysaccharides, lots of different sugars, lots of different combinations that can occur. So when they're actually recognizing antigen, they're looking at those small pieces, right? We're talking about molecular pieces. We're talking about proteins, segments of sugars, sometimes even lipids or nucleic acids. So this is an example of a protein, right? So imagine lots of different amino acids binding, causing this three-dimensional structure. When your body So say this is around, that's molecular weight, I don't know how many amino acids. Fast forward to this one. Okay. So imagine these different sections. These different sections we refer to as epitopes because they're molecularly different from each other, right? What amino acids that are here are different from the ones here, different from the ones here, different from the ones here. So your body could produce an antibody that binds specifically to this one site, right, down to this molecular level, or another one that produces and attaches to this region. Typically, when your body produces antibodies, it doesn't produce just one antibody to the pathogen. It will do like you see here. It will produce multiple antibodies that can bind to multiple different sites on the antigen. These different epitopes, or antigenic determinants. This is referred to as a polyclonal response. Poly meaning many, clonal referring that each one of these antibodies was produced by a specific set of B cells. Because remember I said each B cell has its own unique antibody? So there was a set that were all the same clones of each other that produced antibody number 2. Then there was a whole other set of B cells, genetically similar to each other, clones of each other, that produced antibody number 1. So, I mean, we don't just produce one. We'll produce a whole bunch. But the good news is that they're all going to bind to the same antigen. Now, when we're using antibodies for testing, a lot of times they have made it so that they've purified out just a single antibody. Right? So they're very specific as to what they're binding to. They're actually only using one antibody in the test sometimes. And that's referred to as monoclonal, mono again meaning one. And these are much more expensive to acquire, right? Just a single one. But if we were to take your blood, right, and take out your antibodies from the liquid phase of your blood, for some antigens, as I said, you would produce several different antibodies against the same, say, bacteria or virus. They're just binding at different spots, right? Different molecular regions of that whole huge antigen. Still with me? All right, so when we look at like a bacterial cell, same thing, right? We could produce several different antibodies, all binding each one to a specific molecular interaction with that antigen. So response to antigen varies depending on the type, right? What are you being invaded with? Proteins generally elicit a strong response. Lipids, not so much, more weaker response. Small molecules, if they're too tiny, then we tend not to have an immunological response. So because of that too, when we're certain vaccines and stuff, they actually combine components together to make it a large enough molecule that it's immunogenic, that your immune system will respond to it. So antigenic and immunogenic are used interchangeably, right? They are talking about your immune system mounting a response, right? And although antibodies are the most common way, right, that we can easily detect a response, it's not the only response. So those small interactions, right, so 10 amino acids or so, these antibodies are binding to. So how does a B cell or a T cell have an antibody? And how is it that his antibody is different from someone else's antibody? Well, how do we know, how do our cells know to make proteins? Where is that information stored? In the nucleus. And what molecular molecule? Good old DNA, right? So, how many different, well, we just said, right? A pathogen, like a bacteria, how many antibodies are we going to produce against it? Not just one, right? Multiple ones. Against multiple different sites on that bacteria, on that pathogen, on that antigen. Okay, so how many antigens are out there? Millions upon billions. And do viruses stay the same? Do bacteria stay the same? No, we've already talked about genetics and evolution, right? And this constant change that's happening. So in our DNA, do you think we have all the information to make all the different proteins possible out there? That would be a lot of DNA, would it not? That'd be a lot of DNA. So, that's not possible. So how are we going to make a whole bunch of different proteins all the time, different antibody receptors, different T-cell receptors, if we have a set amount of DNA, and we have the additional problem that even if we were born with all that information, organisms, pathogens don't stay the same. They're continually evolving as we go as well. Anyone ever play the lottery? It's a set number of numbers, right? Just like we have a set amount of DNA. But you can get lots of different combinations, can you not? So guess what our cells do all the time, our B cells and T cells do with our DNA? They basically play the lottery. They take the DNA and they rearrange it and switch it up and do different combinations to create the different antibodies and T-cell receptors. Then we got to check them out, right? Because we want to make sure they don't recognize self. And we're going to eliminate the ones that do. So this is how we generate antibody and T-cell receptor diversity. We do gene rearrangement. They literally pick different pieces of the DNA and put them together, right? Like picking lottery numbers. And also when they pick those segments, they don't always cut them and put them together the exact same way. Right? And I'm going to try and have an activity for us next time to try and demonstrate this, because it's not an easy thing. So how we combine these things, the order and the way in which we combine them, we can make different proteins each time. Except when a B cell does this, it sets this for the most part, this choice. Just like once you pick your lottery numbers, they're set. You can't go, oh, I'm going to change them, right? They're like, no, that's what you picked. You're stuck with them. So the same thing with the B cell. Pretty much once he's picked his general numbers, right, he can tweak it a little bit. But in general, he can't make big changes anymore. And he literally will completely change his DNA, right? And so certain things that you've eliminated, you cannot add back in and switch up your code. So in the DNA sequence, and when we look at, in a moment, how we make antibodies are made of two segments of proteins. A shorter segment we call the light chain, and a longer segment we call the heavy chain. Because guess what? If it's longer, it's heavier. If it's smaller, it's lighter. So when they were taking apart and learning about these molecules, right, they found that you ended up with a piece that was light and a piece that was heavy. And now we know one of them is small and the other one is longer. Therefore, it's heavier. In your DNA is your lottery numbers, and they're arranged in a particular way. There are variable segments, right? And so, for example, there can be between 1 and 300 different ones for them to choose from, right? And then there's the joining segment, and then there's the constant region. So this is the part that stays the same, right? So the antibodies form the same structure all the time. Right? How these two pieces know to come together and join. The longer piece has a diversity region in the center. So you have V, D, J, and then you have a constant region, but we have 5 different classes of antibodies, and the heavy chain actually determines which class. So that's why we have the different letters, mu, gamma, delta, epsilon, and alpha. Right, it stands for the different classes we commonly just refer to as M, G, D, E, and A. So what do our cells do? Because we can't have like tons and tons of DNA, right? Like I said, they're going to play the lottery, right? So they're going to pick their numbers. So this is the DNA. They're literally going to splice out sections and pick just a piece of each one of these sections. They're saying all the stuff I just said. I want to get to the part where they pick. Okay, so there they've picked V2 and J1, right, and put the constant region. This is the DNA in a mature B cell. And then, of course, that can be transcribed into messenger RNA and translated into the protein. And for antibodies, they're going to need to make 2 copies of each. Okay, so you get your 2 light chains, right? And again, these are his numbers. He's picked them. He's stuck. No going back and changing it. Now he's going to do it for the heavy chain. He's going to pick one from each one of the sections. Most commonly, cells are going to start out producing class M, and then they'll switch to the other classes later depending on signals from the immune system. So in this case, though, they're having it go G, which is another common antibody class produced. And so heavy chains, again, bigger, longer, and then they're going to combine 2 heavy chains with 2 light chains. And actually, it'll probably stop right where I want it to. Okay. So, this is a B cell. This is what it chose. This is what it's putting on its surface. And this is also what it'll secrete once it becomes activated and differentiates into a plasma cell. Another B cell, if you were to look at it down to the molecular level, it would have picked different numbers. And even if it picked the same numbers, it's still going to be different because of the way it joins together those sections. Right? So if you have the V segment number 100, it's a segment of DNA, right? A's, G's, T's, and C's. And there is somewhat of a stop and a start point. But even if another cell picked that same number combination, it may cut off part of the end part or part of the start point, right? So it doesn't precisely cut along the same spot each time. Does that make sense to you guys? Yeah, I'm trying to do a paper cutting activity next time. Try and help you guys see what happens. I've been looking and searching on the internet for videos, and none of them show that level of detail. So both B cells, that focused on B cells producing antibodies, which is their B cell receptor. T cells go through this same process, except they just have one antigen binding site, right? So 2 proteins that come together. But they're both in the same in that can recognize a specific antigen, right? So B cell number 1 is only going to recognize a specific antigen and a specific epitope on that antigen. B cell number 2 would recognize maybe a different epitope or a totally different antigen and epitope. Once they have produced these receptors, they're going to be checked out to make sure they function properly. And next time we're going to watch an animation for the T cell. For B cells, this process happens in the bone marrow. And so it makes it easy to remember B for bone marrow, although that's not how B cells got their name, but that's okay. It works. T cells actually did get their name from the thymus, and that's where they go through their maturation process, where they're weeded out if they recognize self or they don't recognize those co-stimulatory markers like the CD markers to be able to communicate with our cells, right? We need to make sure that they can communicate. If they can't communicate, they're not any use to us, and they won't be allowed to continue on. So next time, right, we're going to go through and we're going to label the different parts of the antibody and talk about the different functionalities of the antibody. So if you haven't printed out already, right, this diagram and the others that we'll go through are useful diagrams to print and draw on and color on like I've done here. And then don't forget to read fungi, right? We're going to talk about the fungi case study.